Yes, ladies and gentlemen, I would like to welcome you to lesson 2. The topic is classification of organisms, and we want to discuss kingdom Prokaryote. Remember, in the previous lessons, we looked at the 5 kingdom system of classification, and one of them was kingdom Prokaryote, sometimes called kingdom Monera. So we want to expound. Do you know some of the members of this kingdom? Yeah, anybody who knows any member of this kingdom? Well, I know you got it right. But before we go into the different groups of kingdom Monera, we first want to define what the prokaryotes are. From the two words, pro means before, and then karyon, meaning nucleus. These organisms do not have a true nucleus. Yeah, so they existed even before the membrane-bound organelles emerged. Before the membrane-bound organelles like the nucleus, mitochondrion, lysosomes, chloroplasts, and so on evolved, these organisms existed. And these are organisms whose genetic material is not bound by the nuclear membrane. They have a genetic material, the DNA or RNA, but it is not found inside the nucleus. It is found scattered all over the cytosol, what you would call the cytosol, the general matrix of the cell. They also do not have the membrane-bound organelles, those organelles that are bound by the membrane, like the mitochondrion, the chloroplast, the lysosomes, the Golgi bodies, and so on. They are absent in these organisms. Well, so you'll be wondering, how are they surviving? How do they make up their lives? Well, we are going to look at the details about the prokaryotes. But before we do that, we want to group them into 2 major groups. So kingdom Prokaryote or kingdom Monera is divided into 2 major groups. The first group is Archaea, or sometimes called Archaebacteria. Yeah, Archaea or Archaebacteria. And this group contains organisms that grow under extreme conditions. There are those that are called the halophiles. Those ones grow under extreme salty environments. Then there are those that are called the hyperthermophiles. The hypothermophiles grow under extreme temperature conditions, either in extreme cold, in Antarctica, in the ice caps, and then or in extreme hot, for example in molten magma, when mountains are erupting. These organisms have the capacity to withstand those extreme conditions. So in general, they are called extremophiles. That means they are able to survive under extreme conditions. So those are the Archaea or Archaebacteria, a group of prokaryote kingdom. The second group is Eubacteria. Now these ones, the word Eu means true bacteria. These are prokaryotic organisms, and they are characterized by lack of membrane-bound organelles or membrane-bound nucleus. And they are predominantly unicellular. They only bear one cell, but they have a DNA in a single circular chromosome called a plasmid. And they also have a peptidoglycan on their cell walls. Remember, the plant cell walls have cellulose. Now the bacterial cell wall has peptidoglycan as their major component there. Of course, with other materials, as we shall see later. And they include most of the bacteria that we know, some of which are free-living, others are parasitic, as we shall see later on. And these bacteria are classified into three based on their shapes. There are those that are called the spirilla, the bacilli, and the cocci. The cocci are spherical, they are round in shape, and then the bacilli are rod shaped, they are like sausage shape. And then others, the spirilla, are spiral in shape, like the corkscrew. So we are going to expound more about those shapes a little bit later on. So we now know that bacteria are the smallest unicellular organisms, and they are the most abundant in nature. The weight of bacteria in nature is many times more than the weight of all other organisms combined together. That's interesting, right? The weight of the bacteria is many times more than the weight of all other organisms combined together, meaning they are extremely many and therefore weigh much. But what is fascinating is that they are microscopic. We can't see them. Yes. So let's look at the generalized structure of the bacterium. One is called bacterium, and many are called bacteria. So the bacterium is what you are seeing there. So when you cut it through its structure, its cell, you will be able to see those structures. The outermost part you see is called the capsule, sometimes called the slimy capsule. It's made up of mucus layer, usually for protection. Then the cell wall also there. We said cell wall is made up of peptidoglycan and also other features, as we shall see, the lipopolysaccharides and the rest. We shall see later on. Then we have the plasma membrane, or sometimes in other cells called cell membrane, which is allows movement in and out of cells, selective movement in and out of cells. Then the cytoplasm, an area, a fluid-filled area where reactions take place or metabolic processes take place. Then we have the ribosomes there, and we know majorly ribosomes' role is for protein synthesis. They are sites for protein synthesis. Then we have the plasmid, a strand of where the DNA is found. Then the pili, those small structures there are for attachment, either to surfaces or even to themselves. Some of these bacteria live in colonies, so they use the pili for attachment. And then we have the flagellum, which is used for locomotion or for movement from one place to another. So a much more clearer structure is there, as you can see. The other part we have not mentioned is the mesosome. That part there plays a role. It's a part which plays a role of respiration. It's where respiration takes place. It contains respiratory enzymes. And then the other parts we have already talked about. So that is the structure of a prokaryotic cell or a bacterium cell. Well, let's look at classification of bacteria. Bacteria are classified based on many things. So we are going to see how they are classified. So we are going to look at different aspects on how they are classified. The first way is they can be classified by shape. That is one mode of classifying bacteria. You can classify them based on the shape that they possess. And we have the following shapes. We have the spherical shape. From the word spherical, comes from the word sphere. Sphere means something round. Yeah, and those bacteria that are spherical are called the cocci. Some people pronounce it as cocci. Well, there's no problem as long as you know the spelling. So we have the cocci or the cocci. Those are the spherical shaped bacteria. And one is called coccus. Coccus. Yeah. So they may be in clusters. For example, the staphylococcus, which causes the boils in food poisoning. They may occur in pairs, enclosed by a capsule, for example, diplococci, and so on and so forth. So let's look at that structure there. The first one is called monococcus. Mono means one. So those ones occur in ones, singly. Yeah, they are called monococcus, or many are monococci. When they are in twos, they form a diplococcus. One is a diplococcus, and many are diplococci, meaning they are in twos, in pairs. Sometimes they can form chains, yeah, of more than 3 or 4 long chains. Those ones are called streptococcus. One is streptococcus, and many are streptococci. If they appear in bunches of 4s, they are called tetracoccus, in tetrads, or in 4 groups of 4. If they are in a bunch of very many, they are called staphylococci, and so on and so forth. So, but what is important for us to note is that the word coccus means sphere, spherical. So those bacteria that are round in shape are called the cocci. Yeah, and they can be round and it is only one, monococcus. If there are two, in twos, in pairs, diplococci. If they are in chains, streptococci. If they are in groups of 4s, tetracocci. If they are bunches of very many, staphylococci, and so on and so forth, members. So at least we know now that we have one class of bacteria basing on its shape called a cocci, a coccus, and many are cocci. Those ones are spherically shaped. So we can continue to look at the cocci there. This one is a streptococcus. Those ones you are seeing there are streptococci. They are in chains. But I can also see there. Well, I hope you can now differentiate. You can now tell which one is a monococcus, which ones are diplococci, which ones are streptococci, staphylococci, tetrads, and so on. So our dear students, the second class of bacteria based on shape is the bacillus. Many are bacilli. Those ones are rod-shaped. They are straight. They are rod-shaped, straight. When you look at them in the microscope, they look like some small sausages. That should be looking nice. So those ones are called the bacilli. For example, the common one, the Escherichia coli. You must have heard about the Escherichia coli, usually found in the food. This one is very common and is also found in the guts of humans. And then we have the Bacillus anthrax that causes anthrax in cattle. So those ones are rod-shaped. We can have them also when they are just in ones. For example, we have the bacilli, the monobacilli. If they are in pairs, then you can call them the diplobacilli. When they are in chains, they are also called the streptobacilli. So interesting. So you can have the monobacilli, the monobacillus, or just bacillus, meaning one. If they are in twos, diplobacilli. If they are in chains, streptobacilli. So I hope you can now also identify the bacilli, the rod-shaped bacteria. An example there is the azotobacter. You remember this one in the nitrogen cycle? Yes, it plays a role of fixing nitrogen to the soil, the nitrification process. Then the third class of bacteria, basing on their structure, those ones are curved or spiral in shape. Yeah, curved or spiral, and they can be called the spirilla. You can look at those pictures, they are spiral in shape, curved or spiral, and they are called the spirilla. Then there are some that are curved, like those that are comma shaped. They are like comma shapes, and they are called the vibrio. Yeah, they are called the vibrio. For example, the vibrio cholerae, the one that causes cholera. That one is very common. These days I hear there is cholera in some parts of the country. So that means there is Vibrio cholerae affecting people there. So those ones that are comma shaped are called the Vibrio. Yeah. Then those ones which are spiral are the Spirilla, but they belong to that group of bacteria, curved or shaped bacteria, curved or spiral shaped. Well, then we have the last one there, the filamentous bacteria. Those ones appear like filaments, small filaments, small strips that are coiled. So they are very many and they are also rare. So members, I hope now you can classify the bacteria basing on their shapes. Can you classify bacteria basing on their shapes? Yes. Those ones which are round are called the cocci. They are called the what? The cocci. And there are many types of cocci. They can be monococci, diplococci, staphylococci, and so on. Then those that are rod-shaped, those ones that are rod-shaped are the bacilli. The word is bacilli. One is bacillus and many bacilli. And then examples are there. Then those that are either spiral or comma-shaped, they are called the spirals, or the spirilla. If they are particularly comma-shaped, they are called vibrio. They are vibrio. Then others which are spiral, they are the spirilla. So you can look at that and appreciate the variety of bacteria that exist in nature. So that is one method of classifying bacteria, classifying them based on their shape. Yes, that's great. We want to look at another method of classifying the bacteria. They can also be classified based on their method of respiration. That is the second way in which we can classify them. And in this way, there are those that are aerobic. This one, of course, aerobes are those that use oxygen for their respiration. So these aerobes cannot survive without oxygen. They cannot survive without what? Oxygen. Some of them cannot survive without oxygen, especially the obligate aerobes. But also there are some which are facultative. Facultative aerobes, they can survive without oxygen for some time, but still they will need oxygen at one point. So those are aerobic bacteria. They require oxygen for their respiration. And then we have anaerobic. Anaerobic, they don't require oxygen. They respire without oxygen. And we also have obligate anaerobes. Those ones are even killed when oxygen is there. They can be killed. They are obligate anaerobes. Absolutely do not, oxygen is poisonous to them. Then facultative anaerobes may use oxygen at some point. They can use oxygen at some point, but at some point they may live without oxygen. So members, classification of bacteria by method of respiration. There are two major classes. We have aerobic bacteria, those that use oxygen for respiration. Then we have anaerobic bacteria, those that do not require oxygen for their respiration. That is the second way of classifying bacteria. The third way is by the method of their nutrition, or by the mode of their nutrition. We shall look at them in detail, but we have some which are autotrophic, others which are heterotrophic. So we have autotrophic bacteria. Autotrophic bacteria. These ones manufacture their own food from carbon dioxide. And we have those that are photoautotrophic. It means they carry out photosynthesis. They are photosynthetic. They use energy from the sunlight and to convert carbon dioxide into carbohydrates. Examples include the blue-green bacteria, the sulfur bacteria, and the cyanobacteria. So those ones carry out photosynthesis. They are autotrophic phototrophs. And then we have the chemoautotrophs. These ones carry out chemosynthesis. They use energy from chemical reactions to convert carbon dioxide into carbohydrates. So we have two examples are Nitrosomonas and Nitrobacter. We have two autotrophic bacteria. We have photoautotrophic that use light. Then we have chemoautotrophic. Those ones use chemical energy. But all of them are autotrophic bacteria. They can make their own food. Then we have the second one, heterotrophic bacteria. These ones cannot manufacture their own food. So what do? They feed on already made organic food, and they feed in different ways. For example, we have the chemoheterotrophs. These ones obtain energy from chemicals in food. For example, we have the saprotrophic bacteria, obtain their food from dead and decaying organic matter. Saprophytism is a process of feeding on dead, decaying matter, and majorly it is bacteria and the fungi that do it. The bacteria that do it are called saprophytic bacteria. So what do exactly? They secrete enzymes into the food, and then after that those enzymes break down the food molecules into soluble products, after which the bacteria absorb these soluble products and then assimilate them into their bodies. Those ones are saprophytic bacteria. The other type of heterotrophic bacteria are the parasitic bacteria. From the word parasites, these are organisms that live in other organisms called hosts. They obtain food from them, but also they cause harm to them. And these ones include most of the bacteria that cause disease. Those ones are parasitic bacteria. They cause harm to the host or the host cell. Then others are mutualistic bacteria. So these ones particularly live in close association with the legumes in the root nodules. An example of a mutualistic association is that one that takes place in the root nodules between the rhizobium bacteria, or what we call the nitrogen fixing bacteria, and the leguminous plant, where the nitrogen fixing bacteria participates in the process of converting atmospheric nitrogen into usable nitrates by the plants. So plants will get nitrates from the soil that have been converted by this bacteria, and then the bacteria obtains nourishment and protection from the root nodule of the plant. So all of them benefit. So bacteria can be saprophytic, they can be parasitic, they can be mutualistic. But as long as they don't make their own food, they will be called heterotrophic bacteria. And that's the second group of bacteria basing on the feeding. Yes, there are many other examples of mutualistic bacteria, including the Escherichia coli that produces vitamin B and K, and others as well. So, another way of classifying bacteria can be based on staining reaction of the Gram stain. That stain was developed by a man called Gram. So the stain is called the Gram stain. And there are two. The first one is the gram-positive bacteria. They stain purple with the Gram stain. The cell walls lack an extra outer membranes, the lipopolysaccharides. So members, in this staining process, after staining many bacteria, it was noticed that some bacteria stain is purple, others stain pink. The original color of the Gram stain should be purple. So those that respond, that stain purple, are called the gram-positive bacteria. They stain purple because they have a bigger layer of the peptidoglycan that forms the cell wall of the bacterial cell. The peptidoglycan layer is very large, it's enlarged, and can retain the purple color of the gram stain. So those are called gram positive. But they are also disadvantaged because they lack other extra layers. They only have a bigger peptidoglycan layer which retains the gram positive, the gram stain, but they lack other layers that might have been good for them as well. For example, now in the gram negative bacteria, these ones stain pink with the gram stain. It means that their cell walls have a very thin, their cell walls, I want you to listen properly, their cell walls have a thin peptidoglycan layer that cannot retain the purple color. So the purple color fades out, and then you will be able to see the color pink. So that is the gram negative. So gram negative stain pink with the gram stain, then gram positive will stain purple. But also the gram negative are advantageous because they have other extra outer membranes which are made up of lipids and polysaccharides. Those ones are called the lipopolysaccharide layers, and those membranes are good for protection. They protect the bacteria against the antibiotics, for example penicillin. So if you are a doctor or if you are a chemist, which bacteria would be easier to destroy, gram positive or gram negative? Well, I leave that to you. So based on that, you see that the gram negative have extra layers that makes them defensive. So bacteria can also be classified based on their response to the staining reaction. You classify them into gram positive and gram negative. The gram positive, those ones stain purple with the gram stain, they retain the purple color, and then the gram negative do not retain the purple color, and they are seen as pink. Okay, so that's great. So we now know how to classify bacteria based on different aspects. Now, having known about that, we can now look at the population growth curve of bacteria. Bacteria can be grown even in the laboratory. It is called culturing. You can culture bacteria even in your lab and then monitor their growth over time. You monitor the increase in number over time. So from here you can see that graph. It looks like an S. It's an S-shaped curve. That kind of curve is called a sigmoid curve. You can see there A, part B, C, D, and E. So we want to explain those parts. Let's start with part A. It is showing us the population growth against time. You do not see population growth there, but yeah, it is population growth against time. So that part A sometimes is called the lag phase. The lag, yeah, lag, L-A-G, lag phase. So according to, when you look at it properly, you realize that the population increases gradually because the bacteria are still adapting to their new environment and the growth has not yet achieved the maximum rate. The bacteria also are still releasing new substances and still adapting to the conditions. That is why their population is increasing very slowly there. Yeah, in part A. Then part B, you can see there's a great increase. The population increases rapidly with time. The bacteria have adapted to the environment and the enzymes have been synthesized to digest the food which is in abundance so as to support the rapidly increasing population. That's what you are seeing there. Rapid increase because of availability of food, availability of reproducing species. That's why you see rapid or even exponential growth. Then when you look at part C, there is a slight decrease in population there. It increases with a decelerating form, in a decelerating fashion, simply because probably this bacteria, the resources are getting depleted. The food, the oxygen, and so on are no longer enough because of the high number of bacteria present. So there's a lot of competition there and because of that, the rate at which they are reproducing is reducing. That's why you are seeing there is a decrease there. There is a slight decrease in the way it's increasing. Then part D, you can see that there is a slow population growth, or even you can see from there that largely there is even a constant population. There is no increase, no decrease. So what can explain that? So what is clear is that if the population is not increasing and decreasing, it means that the number of organisms produced is equal to the number of organisms dying. The death rate is equal to the birth rate. That is what you can see there, making that curve flat at that point. And then beyond part D, you can see part E. Now in that part E, what you can see is that the population declines slowly because the cells stop multiplying due to exhaustion of oxygen, accumulation of wastes, and even exhaustion of nutrients. So at part E, you can see that there is a decrease there that you can see. There's a very big decrease in the population, or a slight decrease because of the factors we have mentioned there. Well, that is what you can see there. We have already explained that. So let's look at factors which affect bacterial growth. Yeah, which factors are affecting the increase in population of bacteria? One is nutrient availability. Of course, when nutrients are available, the bacteria will grow faster. Their population will increase. When nutrients are not available, then the population will not grow faster. The second one is oxygen availability. For aerobic bacteria that use oxygen for respiration, increase in oxygen presence or availability favors their population growth, and vice versa. Decrease in population in oxygen also decreases their population growth. But for anaerobic bacteria, presence of oxygen poisons them, especially the obligate anaerobes. The oxygen is available, then for them it is poison, and they will not increase in number. Instead, they will die out. The other factor is temperature. High temperature speeds up bacterial population growth up to a temperature of about 40 degrees Celsius. That is now the optimum temperature. So beyond which there will be decline because the enzymes will be denatured. The increase in population growth is as a result of activation of enzymes. As the temperature increases, enzymes are activated and they are able to divide faster. But also very low temperatures slow down the bacterial growth because enzymes are inactive at low temperature, you know that. So that is how temperature affects bacterial growth. The other one is moisture, availability of bacteria grow in moist and warm places. That's why you look at your body, under the armpits, between your legs, and other moist and warm areas have a lot of bacterial growth. Even in your mouth, when you cover your mouth for so long, there will be some foul smell, simply because it's a favorable condition for bacterial growth. Moist and warm environment. Okay. The other one is accumulation of toxic wastes. For example, carbon dioxide. Carbon dioxide lowers the pH of the medium, and that does not favor bacterial growth. Okay. Thank you very much. At least now we know factors which affect bacterial growth. We know nutrient availability. Can you explain that one? Oxygen availability, temperature, availability of moisture, and then accumulation of toxic what? Waste products. So our dear students, thank you very much for being very good. So at this point, we want to look at economic importance of bacteria. Now, the word economic importance, who can tell us? Okay, Kemba is saying that, Second, what are you saying? Well, Timothy, Jacqueline, well, all of us agree that economic importance is either advantages or disadvantages. I think I agree with you. So when you look at economic importance, we are looking at advantages and disadvantages of bacteria. So one, they are cultured for research purposes. There's a lot of research in therapeutics, in pharmaceuticals, in microbiology. There's a lot of research that takes place using bacteria. And if bacteria were not there, it would be so hard to research, especially on microbes. So that is one advantage. Two, they facilitate the making of foods like yogurt, cheese, and vinegar. I know most of you enjoy yogurt, yes? The ladies out there, you enjoy yogurt, I know. But do you know how it comes? Do you know how it is made? Well, at least now we know that it is the bacteria that play a part in coagulating of this milk products to form yogurt, which is good. It has a commercial value. So if they had not been for bacteria, it might have been difficult or even expensive to make yogurt. So you can see there the process on how yogurt is made. The other one is it's used in making antibiotics, amino acids, and enzymes. So bacteria, after research of microbiology and therapeutics, bacteria is used for manufacture of antibiotics. They are cultured in large numbers, and then they give us some antibiotics that we use for treating different ailments. The other one is in humans, vitamin K and vitamin B complex are produced by symbiotic bacteria called Escherichia coli, which is located in the gut, in our digestive system, in the intestines. So that one produces vitamin K, which is a good clotting factor, and then the vitamin B complex, a very good immune booster there, and also plays a role in very many metabolic activities in the body. They cause decomposition of dead organic matter, hence enabling disposal. Yeah, might have seen different organisms after dying, they rot and decompose. So bacteria are the ones that are responsible. Bacteria and fungi, those that feed saprophytically, are the ones that are responsible for decomposing this organic matter. And then in the process, they take part in nutrient recycling as well. In addition to decomposition, they also fix nitrogen to the soil. We already talked about that earlier on. We have rhizobium bacteria, for example, that play a role in converting atmospheric nitrogen to usable nitrates. The other one is they cause food to get spoiled, to go bad. You must have seen your tomato going bad, or your food, or your good food that you have cooked. In the morning, it is smelling badly. So what could have happened? As you are sleeping, there is a lot of business taking place by the bacteria to make that food go bad. As they feed on it, they also deposit wastes there that make the food unpalatable. And when you eat such food, you suffer from food poisoning. So some bacteria and sulfur vibrio produces sulfuric acid, which destroy underground pipes. These are bacteria that live in the soil. And those metallic pipes that are, these days we have few metallic pipes. But if they are there, they can get degraded by the acids that are produced by the bacteria. So our dear students, thank you very much for being very attentive in this lesson. And I know you have learned a lot about bacteria. I want to just encourage you to make a summary of what we have learned. Yeah, but also to read further and expound more details about this kingdom Prokaryote or Monera. Thank you very much. May God bless you.